I'm Kira Curtin, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Next Generation Sequencing of Immunoglobulin Genes in Lymphoid Malignancies, and is sponsored by InVivoScribe. Our speaker today is Rena Xian, an Assistant Professor of Pathology and Oncology at Johns Hopkins School of Medicine. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Rena. Rena?
Okay, good morning, afternoon, evening, wherever you may be. My name is Rena Xian. As the title of the slide has shown here, over the next 45 minutes or so, I'll be talking to you about next-gen sequencing of the immunoglobulin genes in lymphoid malignancies, particularly B-cell processes. So we'll be mainly focusing on the B-cell receptors. The only disclosure I have to give that's relevant is actually today's talk. As we had said, this is sponsored by InVivoScribe. To begin, here are our learning objectives, right? So I will begin with some background on the actual targets that are being sequenced. Then we will compare sequencing techniques that are available. Then I will talk about some pre-analytical variables that you should be thinking about if you're thinking about adopting this for research or clinical use. And then lastly, I will compare, contrast sort of how you determine clonal proliferations by next-gen sequencing versus the more traditional techniques. And then the one sort of disclaimer I want to put on all of this is that any data I share, this was generated from my research work, and it's not clinical testing. Okay, so let's begin. As many of you know, if you're here, you must be interested. As many of you know, B and T cells are critical to adaptive immunity. Their receptors and the interaction of those receptors with each other and immune cells is what allows for all of us to have a robust immune system that can recognize, that can respond, that can suppress infections and foreign agents. Now, instead of attempting to encode all of the genes necessary to recognize every possible antigen, our genome actually encodes parts of genes, what are called variable, diversity, and joining genes. These become modules that can then be recombined in sort of endless ways, right, to form a complete B or T cell receptor. So on the on the left, you can see the left panel right here. You can see a number of known functional and non-functional genes within these individual modules, the VDJ genes, that encode either a full complement of B or T cell receptors. This shows you that the variable genes are usually the most numerous, which is followed by either the diversity or joining genes depending on the locus. This also demonstrates that certain genes only contain VJ combinations whereas others contain VDJ combinations. Now on the right, this is focused more on the B-cell receptor. So now these are grouped by heavy chain and the two light chains, which are kappa and lambda, and they are grouped into what we call V families. These are gene families that have a lot of homology among one another, but there are discrete numbers of genes within them. You can see based on the functional and non-functional, again, distribution, that some V families have a lot of functional genes, such as the V3, the kappa 1, right, the lambda 3, but other families are quite small and some don't even contain any functional gene members. So what this tells you is that if there are abundant V genes within a family, then those V genes are more likely to be selected for recombination and ultimately more likely to be expressed. Now let's focus on the IG genes a little bit more. So this is the same panel as the one on the right-hand side, but now enlarged. And now you can see above the original figure, there is a table. So what does this table represent? So this table represents the number excess, the fold excess of potential germline polymorphisms or haplotypes that exist for a particular V, D, or J gene, and even C gene. So these numbers represent the alleles that are present in the population that we know of. So for example, if you look at this IGHV gene, there are approximately 50 known reference functional V genes. And this 5.5 means that there are about 5.5 times more alleles of those V genes. Then we have approximately 300 or so functional V gene haplotypes that are known. So these data were illuminated through next-gen sequencing, and in recent years, these sequencing efforts have shown that the Ig locus, particularly IgH, may actually be among the most polymorphic loci in the human genome. So in fact, if you think about diversity of antibodies or receptors, there is a, within individual diversity of the VDJ genes, that are selected. However, there's also a population level diversity that reflects the individual's own V and D and J genes that they've inherited, which contributes to what I call a population diversity. So given this population information, we believe that different groups carrying different germline haplotypes, that can often show not just single nucleotide variants, but also even deletions, duplications, or entirely new V genes altogether. So we believe that this population diversity contributes to different populations forming different antibodies that may recognize the same pathogen as this cartoon shows you on the right, but perhaps they recognize different epitopes or you have different neutralization abilities, and that these host genetic differences may actually contribute to how individuals respond to the same antigen and therefore potentially same vaccine. So knowing this, the components that contribute to make B cell and T cell receptors, let's talk about how they're formed. As mentioned earlier, these genes are modular. They're brought together on the genomic DNA level, and they rearrange to form the functional receptors. However, in non-B or non-T cells, these genes actually remain in their germline configuration, in this configuration at the top, where all the individual modules are spread out over megabases, so that only lymphocytes, either a B or T cell, will have recombined receptors. So in these lymphocytes, early in lymphocyte development, BDJ or VJ recombination is mediated by RAG. Sometimes even before B or T cell fate is determined. In these loci that have V, D, and J genes, the rearrangement process generally occurs first with a DJ rearrangement, as is shown here, which is then followed by a V to DJ rearrangement. So this occurs in both the IgH heavy chain and the TCR beta chain. This now brings together a single VDJ, in close proximity. And then additional nucleotides can be added at the junction points. And then after recombination and transcription, it's through slicing that the final mRNA of the final receptor is able to be formed. So one thing to note is actually you see these circular excision structures that are present. So these actually represent the intervening sequences from that original allele that are left over after the VDJ recombination has occurred. These intervening sequences are essentially lost forever from that cell. And that allele actually is unable to participate in future rearrangement events. And this is important as we think about how hierarchical the rearrangement events are. So not only does DJ rearrangement always precede VDJ in the setting of a VDJ gene, this entire process of B and T cell development is coupled, highly coupled to the rearrangement of the B cell and T cell receptors. Now, if we focus on B cell for this moment, the DJ occurs usually in the pro-B stage. It will continue to a VDJ throughout, usually in the pre-B and throughout the rest of the maturation process. At this stage, the cell may have a functional heavy chain, but it will not yet have, you see at this stage, it will not yet have a functional light chain. And the light chain rearrangement usually begins with the VJ rearrangement of the kappa gene. You can see here, kappa begins usually in the pre-B. Now, if that's successful, that B cell will then become a kappa-expressing B cell. However, if unsuccessful, that allele that generated the non-productive kappa rearrangement may engage what we call the kappa deletion element, which prevents that allele from ever being used again. Now, this KDE, what we call KDE rearrangement, could signal the rearrangement of the alternative allele that hasn't been used, the alternative kappa allele, or it can actually signal the subsequent rearrangement of the lambda locus. So a successful lambda gene rearrangement will rescue that B cell from apoptosis and therefore allow it to develop into a lambda expressing B cell. So looking at the potential rearrangements that could be detected on the DNA level, you can see that all lambda expressing B cells should harbor evidence of that prior kappa deletion element event from the prior unsuccessful kappa rearrangement. So in an analogous way, T cell receptor is also rearrangement is also highly ordered and regulated during T cell development, but we won't be discussing that today. What we'll move to now is to look at the rearranged VDJ gene structures, which are actually very similar even in the absence of the diversity genes in some of the loci. So if we look at the organization, you can see that either on the gDNA level, genomic DNA level, or the cDNA level, the genes are arranged with similar structures. There are what we call leader sequences in yellow. There are what we call CDR, or complementarity determining regions, in the color barcodes. And then there are intervening framework regions in green, which is followed by the constant regions in gray. So once on the genomic DNA level, we had said that once a VDJ rearrangement has occurred, the intervening sequences are removed and they are brought to close proximity. However, you can see that the full leader sequence is not brought into proximity until the mRNA is formed through splicing. So why do all these genes, whether you're IgH, Ig kappa, TCR alpha, so why do all these genes have the same structures? Well, this relates to the ultimate function of the peptides that are encoded. So the CDRs, as many of you may know, they actually form the loops that ultimately display on the surface of the antibody or the B cell receptor, and that's where antigen recognition occurs. Whereas the framework regions encode the amino acids that form the core beta sheets of the receptors, and they essentially function as scaffolds. So these scaffolds are purely structural in nature and do not typically contribute to antigen specificity. So for this region, the CDR sequences are usually favor for somatic hypermutation and are very different from one B cell to another, whereas the framework regions are not only not favored for somatic hypermutation, but are relatively conserved. And so this is actually a perfect segue into the next portion of our talk, which is comparing sequencing techniques. So knowing the structure, which is predictable, of the different VD and J gene rearrangements, we can actually think about how to design a sensible assay. So since genomic DNA and cDNA configurations of the Ig receptors are somewhat different, different strategies may be applied, whether you're starting from DNA or RNA. So let's start from DNA first. If we look at the IgH gene as an example, you remember the leader sequences are not, or the upstream leader sequences rather, are not brought into proximity until splicing, so they are still quite far apart. However, what you do have in proximity are the V and potentially D, if that D is being encoded, VDJ genes. Since the framework regions are relatively conserved, most amplicon-based sequencing techniques will have primers that target the framework regions, either a family, right, so conserved families, through family framework primer sets, such as was used in the original Biomed 2 design, or there can be V gene specific primer sets that are brought together in a multiplex reaction so that every single V and J combination can be recognized. Next, you can target the closest, the proximal leader sequence in a similar fashion to the V families by having multiple leader sequences that can amplify to a common J primer. And then one advantage of genomic DNA is that even non-productive alleles, so remember before we had talked about DJ rearrangements precede VDJ. If that initial DJ rearrangement is unsuccessful, that allele is actually aborted as an incomplete rearrangement. So the evidence of that DJ allele could actually be recognized on the genomic DNA level and could be detected with non-coding sequences upstream of the DJ and overlapping with the D gene itself to amplify these very small incomplete rearrangements. However, whatever combination of primers you use, these are always going to be highly multiplexed. And so these sequencing strategies can actually suffer from amplification bias, depending on the degree of sequence homology among the primers and the CG content of your template. So one way investigators have sought to avoid this problem is actually to start from RNA. So the same framework-based approach can be used, but then you're still at risk for amplification bias. So the other way to avoid amplification bias is to adopt a completely different design. So to reduce amplification bias, you have to reduce the complexity of your primers and or probes, which can only be done on the RNA level because in this situation, you have a complete leader through constant region amplicon that can be treated like any other RNA fusion. So those of you who are familiar with RNA fusion sequencing, this essentially becomes a problem of a known gene with an unknown partner gene. So the known gene, quote unquote, in this case, could actually represent the conserved J primers or the conserved constant region primers, which serves as the anchor, as the gene-specific primer. Then the other side is either amplified with a poly-T primer after poly-A tailing, which is a technique called 5' rates, or after ligating on universal primers, as shown in the cartoon here, that is akin to anchored PCR. So various methods have been developed to sequence the IG genes or TCR genes using RNA based on this type of principle. Now, whether you choose DNA or RNA, you must consider the different possibilities, the pros and cons. So let's look at them. So DNA obviously is more stable in whatever sample type you start with. But you could argue that RNA is more abundant since the number of copies of RNA is not limited by the number of alleles that have been rearranged. However, all alleles, productive or non-productive, can be detected by DNA up to 4 if you're looking at the kappa locus, sometimes even more, whereas RNA techniques will only allow you to detect a single productive allele. So not only does this make the argument that DNA offers more potential targets, this also shows the downfall RNA techniques that may not detect clonal sequences when there is either no productive rearrangement or the B-cell malignancy is too early in its life to have a productive rearrangement, such as lymphoblasts. So next let's consider the sequencing techniques. So I mentioned earlier, amplification bias can be substantial in DNA assays, whereas the bias is typically minimal in RNA assays. Another side effect of too many multiplex primers used in DNA assays is that you're more at risk for mispriming events or leading to sequencing errors. This is because there are more introns, there are partial Vs, there may be retained genes that could interfere with actual amplification of the target itself. So the last set of considerations relate to diagnostic and analytical sensitivity for clonality determination and clonal quantification. Since RNA is limited to a single productive rearrangement per cell, DNA is typically preferred for improving diagnostic sensitivity because of the ability to recognize non-productive alleles. Now, analytical sensitivity is, however, limited by the number of rearranged haploid genomes, which obviously cannot exceed 2 per cell. While analytical sensitivity RNA may be superior because the message could be much more abundant. Now, obviously, in situations where there's a non-productive allele, the same argument cannot be made because there will be no message RNA. And lastly, regarding quantification, the ability to count the number of genomes is much more realistic for DNA since you know the number of genomes in a cell, but for RNA, it's actually quite difficult because of that excess of message when there is a potential increase in message, but not on the DNA level. So considering all of these things, the field has really become divided in two. The DNA-based approach has been adopted, more or less adopted for clonality determination for lymphoid malignancies, both for diagnosis and for monitoring, whereas the RNA approach, as you can imagine, because you only want to see productive alleles, RNA has been adopted by the immunology community to look at repertoires. So those are the major differences in the two nucleic acid analytes. And for the remainder of this talk, I will focus actually on DNA-based approaches. So let's review a few available DNA NGS techniques. And we can compare this to the original Biomed2 PCR-based technique. So there are three available techniques out there currently. One is based on the, all of it is based on the original Biomed2 primer design. However, only one, this Euroclonality Biomed2 group, involves the same consortium that assisted with designing Biomed2 to begin with. So you can see the Euroclonality group's NGS assay and the in vivo scribe NGS assay both employ consensus framework region primers that amplify across to a J gene primer. Differences there is that the Euroclonality group has chosen only to have a framework 3 to J amplicon and a DJ amplicon, whereas the in vivo scribe assay that we have used in our lab will look at both frameworks 1, 2, and 3 together without DJ. Now, the adaptive or clonal-seq, as it used to be called, is slightly different because it does not use consensus V gene framework primers. It will have a specific V for every V framework it has in its database, and it uses those to amplify individual VJ combinations. So there are subtle differences in the design, and so do these subtle differences actually affect diagnostic sensitivity? And the answer is probably and somewhat, depending. So let's compare to the original PCR technique as the gold standard. The NGS techniques overall, if you look at maximum sensitivity, overall actually show quite similar sensitivities, though disease-specific sensitivity are not as well established. And the primer set level sensitivities are not always comparable. The adaptive technology, as you may know, is the only FDA-approved sequencing assay for minimal residual disease monitoring. In the diagnostic setting, their sensitivities are very good, but trend lower, which may actually be related to the fact that they are sequencing a lot of multiple myeloma tumors, which actually are post-terminal center and maybe have more somatic hypermutation and chance for target dropout. The Euroclonality assay does approach a sensitivity of 96% in a variety of non-Hodgkin lymphomas. And remember, they only use the framework 3J and DJ primers alone. The LymphoTrack assay actually reaches a quite high sensitivity with just one framework. However, combined with LEADER, it can achieve up to 97% sensitivity. So what you should take away from this slide is that no matter the NGS technique, the diagnostic sensitivity is more or less comparable to standard PCR techniques, and that the real power of NGS lies in its ability to not only find a clone, but to find the sequence. So in the next part of this talk, I want to give you a preview of some of the data that actually are generated from LymphoTrack-based sequencing. With any new technique, you want to know what baseline looks like. And in this case, you want to know what polyclonal looks like. And it's not as straightforward as you might think. So the IGH assay from LymphoTrack contains 3 amplicons targeting framework 1, 2, and 3 regions of the variable gene, very similar to the Biomed2 design. Shown here are aggregate data from 4 polyclonal samples, where the amplicon length is displayed on the x-axis and the raw total read count is displayed on the y-axis, and each dot represents a single amplicon. You may be used to seeing capillary electrophoresis displays of PCR, and so this actually mimics that in the way that there is a rough Gaussian distribution, whether you're looking at framework 3, which is the smallest amplicon, or framework 1, which is the largest amplicon, so that each group does, the amplification result does approximate a Gaussian distribution, just like you would have with PCR. And you can see I tried to color code the different V families, and it does appear, right, just like the traditional PCR technique, that every framework, that every V gene is amplified 3 times by every framework primer. And so to convince you that that's true, let's look at a breakdown. If we break this down into the component V genes from 1 through 7, you can see that the dots are represented for each V gene more or less by every framework primer set. Additionally, you can see that the V3 and the V4 family sequences are most abundant, which is more or less related to the fact that these families have the largest number of V genes within them. So next, I want to move on to Ig-kappa. However, before we can look at Ig-kappa data, I should review how kappa rearrangement occurs, because there are intricacies there and it is different from the heavy chain rearrangement. So unlike Ig-H heavy chain, the Ig-kappa gene only employs a V and J combination. Now, the first VJ rearrangement, as you can see here, may be functional, in which case that B cell will become a kappa-expressing B cell. However, when that rearrangement is non-functional, instead of then rearranging the next allele, just like IgH, there's actually an additional step. This kappa deletion element I alluded to earlier will be engaged. This kappa deletion element rearranges to actually prevent that allele from ever being used again. And it can actually rearrange to either an upstream V, right, in which case the intervening non-functional original VJ rearrangement would be removed, or it can rearrange to an intronic signal, thereby keeping the original non-functional VJ intact. So in the first scenario, you will see a single clonal sequence that represents the KJ, KDE, and J rearrangement. In the second scenario, you will see 2 clonal sequences, one representing the non-productive VJ and the other representing the intron KDE. So in both scenarios, a single allele is affected, remember, a single allele. And once this kappa allele is deemed non-productive by engaging KDE, it will signal either rearrangement of the second allele, in which case this can occur again twice, or it will signal rearrangement of the lambda locus. So now that you have a sense of the possible combinations of rearrangements, let's look at the data. So if you remember, all B and T cell receptor genes have the same framework configurations that flank the CDRs. So you can see here, again, similar to the Ig heavy chain sequences, the amplicon length is displayed against the read count of each individual amplicon, shown as dots. And you can see that the smallest framework is framework 3, the largest framework is framework 1, and then again, the distribution of the dots approximates the Gaussian distribution. So the question now is, do the IgK primers target every framework of every V gene, just like Ig heavy chain? And I will show you that. The other question you should be asking at this point is, why are there amplicons that are both larger and smaller than the known framework regions? So if we do the same component breakdown and we look at VJ rearrangements first, we see that unlike Ig heavy chain, not all V families are amplified equally by all primers. For example, V kappa 1 is mostly amplified by framework 3 and framework 1 primers, but V kappa 3 is almost exclusively amplified by framework 3 primers, and V kappa 4 is almost exclusively amplified by framework 2 primers. So not all frameworks will be not all genes will be amplified 3 times in this assay. Next, we can look at the kappa deletion rearrangements. They are displayed here as either VKDE rearrangements or intron KDE rearrangements. And we see that the majority of the KDE rearrangements actually fall within what we believe are framework region amplicons. So they overlap with the framework region amplicons. But what we do also see is that occasionally they will be much, much longer than the existing framework amplicons. And you can see here that occasionally they will overlap with 2 framework amplicon regions, such as the kappa 3 KDE, And then lastly, I want to point out that the intron KD rearrangements are typically fixed in length and typically resolve with the largest framework 1 amplicon. So seeing these distributions of the normal polyclonal sequences, I hope you appreciate that there isn't redundant sequencing of the kappa genes as there is of the IgH genes. However, the sensitivity of the kappa locus is not inferior and is because of the abundance of KDE sequences that are still able to be detected. But with kappa, interpretation should be much more careful because there could be multiple alleles rearranged, multiple KDE versus VJ rearrangements that could still represent a single allele of a single neoplastic population of cells. So we've talked at length about the amplicon length. And obviously, the more times you're able to interrogate a single rearrangement, i.e. through redundant sequencing through framework 1, 2, and 3, the higher the likelihood of identifying a clonal sequence. And this is why IgH sequencing is redundant. However, there may be amplicon dropout, and this is typically related to somatic hypermutation, which is a normal part of B-cell development and a characteristic of both germinal center-derived and post-germinal center-derived neoplasms. The next question you should be asking is, could there be amplicon dropout for reasons other than somatic hypermutation, i.e. could there be other reasons for reduced sensitivity? And so the question to ask now is, we should think about the input DNA integrity. Is the integrity of the DNA sufficient for assessment of all loci? As many of you know, the traditional PCR technique has a sizing marker associated with it to inform you whether appropriate lengths of DNA fragments are even present in your specimen. But these QC metrics, these QC primers are currently not available for the NGS techniques. So you have to then think about the pre-analytical variables and how you would control for that in your own lab. So we'll next talk about the pre-analytical variables. But before we expand, I want to give you a sense of the expected amplicon lengths by sequencing as compared to PCR. As you can see, the NGS sequence itself, the actual rearrangement itself, appears smaller when you look at the data. And that is because the primer sequences are trimmed. So they're not part of the final clonal sequence designation. However, if we add approximately 50 bases to the amplicon lengths, to the sequence, the rearranged sequence, we see that these amplicon lengths are approximately the same as the current PCR product amplicon lengths. And that the largest framework 1 amplicons require molecules that are at least 300 bases in length. Now, 300 bases is easily achieved when starting from DNA isolated from fresh specimens, fresh cells, whole blood. However, this is not as straightforward when dealing with formalin-fixed embedded tissue that many of us use on tumor tissues. So are the DNA lengths isolated from formalin tissue sufficient? If 300 base pair is a minimum bar to meet for framework 1 amplification, if you look at the actual distribution of lengths of isolated DNA, this Mayo group showed really nicely in a recent paper that the recovered DNA length from FFPE actually varies by extraction method and also varies by tissue type. So what they did in this study is they performed gDNA isolation using 9 different isolation kits that were either manual or automated, and they performed this on 3 different tissue types, breast tissue, pancreas tissue, and brain tissue. These pie charts then reflect the proportion of fragments of DNA that were either 1 to 200 bases, 200 to 201 to 1,000 bases, or excess of 1,000. Remember, 300 bases, right, is our minimum for framework 1. So the interval needed for framework 1 and 2 amplicons actually lie in this dark gray or black. So what these data tell you is that irrespective of extraction method, cerebellum or brain tissue almost never yielded DNA in excess of 1,000 bases. but that at least half of the DNA, typically half of the DNA, or between 200 and 1,000, therefore should be sufficient for IG sequencing. Now, for breast and pancreas tissue, this group showed that certain extraction methods had a greater effect on recovering larger DNA fragments, whereas others were not. So this means that DNA integrity and adequacy could vary by the method of DNA extraction you use in your lab and could vary by tissue type, and certainly should be a consideration when thinking about doing IG sequencing downstream. Now, another tissue type or source that is very challenging for IG sequencing is plasma cell-free DNA. There is a growing literature in the past few years suggesting IG sequencing is a sensitive marker for treatment response monitoring, i.e. minimal residual disease monitoring in patients with aggressive B-cell lymphomas. These lymphomas form masses, they don't circulate, so it's actually very difficult to assess response. Despite the fact that plasma is a fresh whole blood derived specimen, plasma DNA is highly fragmented. And we did a study a couple of years ago, that a feasibility study of IG sequencing in plasma, and we showed that plasma cell-free DNA collected from patients with lymphoma or without lymphoma, the plasma DNA integrity was typically sufficient for framework 2 and 3 amplicons, but were not always sufficient for framework 1 amplicons. And what these graphs show you is amplicon length on the x-axis, and then proportion of the reads that occurred at each amplicon length, proportion of all reads on the y-axis. So you can see again the overlay with a polyclonal control in gray, that the framework 2 and framework 3 profiles overlap quite nicely with genomic DNA control. However, the majority of framework 1 amplicons were actually much smaller than expected. And when you looked at the sequence behind those shorter amplicons, you realize that most of them often did not represent complete VGJ rearrangements. Rather, they represented partial V genes or incomplete VG combination partners, suggesting non-specific amplification when larger DNA fragments were missing. So looking at these data, some might say, well, framework amplicons are simply not informative for cell-free DNA and should not be used. However, when we summarized our clonal versus non-clonal data, we realized that there were certain lymphoma patients in whom framework 1 and 2, such as this patient here, framework 1 and 2 primers actually were the only primer sets to yield a clonal signal. So that framework 3 alone approaches are likely not enough for sensitivity purposes. So with these data, we actually sought to see if we can improve the recovery of larger cfDNA molecules that might improve the sequencing yield of framework 1 amplification. So before we discuss our efforts, I want to briefly introduce cfDNA or cell-free DNA and its constituents. So when whole blood is spun down, it separates into 3 compartments: plasma at the top, where cell-free DNA is found; white blood cells or leukocytes in the middle. This is where PBMCs are usually recovered, and red blood cells at the bottom. On the right panel, you can see the different cell types that contribute to cell-free DNA in healthy individuals, as identified by this Israeli group a few years back, where they did methylation profiling of cell-free DNA and therefore assigned a cell of origin. And they showed that the majority of cell-free DNA is actually derived from circulating leukocytes, shown in blue here. And the reason for this is that cell-free DNA reflects cell turnover and apoptosis, at which point nuclear DNA becomes fragmented into discrete units that approximately counts up as a single nucleosomal turn. Sometimes there is linker associated with it, but we call these cell-free DNA molecules either mononucleosomal, i.e. one single unit and turn, or dinucleosomal. Occasionally you will see 2 doubling of the length. The mononucleosomal molecules are typically around 150 or 160 bases, which is far below the amplification requirements for even framework 2, not to mention framework 1 amplicons. Now, does this cell-free DNA length actually result in shorter or larger amplicons. So this group from Canada showed a couple of years ago that the insert size, this is a hybrid capture-based next-gen sequencing assay rather than amplification-based, but it's a very similar concept. They showed that insert size estimated from NGS was larger when you had more prominent dinucleosomal molecules. So on the left-hand figure is actually a high-sensitivity gel electrophoresis of the cell-free DNA itself. The blue tracing shows you a typical isolate where the mononucleosomal molecule, right, that big peak is the predominant population of molecules, whereas this dinucleosomal peak is very small. Whereas the second patient here, you can see in red, the mononucleosomal molecule is slightly bigger, but likely not significantly bigger. However, what's really different here is that the dinucleosomal molecules are much more abundant. And that's actually reflected in the lengths of the inserts from hybrid capture sequencing. On the right panel, you can see that the blue tracing corresponds to much lower, more abundant short fragments in sequencing, whereas the patient with the more abundant dinucleosomal molecules had larger insert sizes. So knowing that cfDNA length likely dictates the sequencing read length, our group set out to see if longer cfDNA molecules could be enriched during the DNA extraction process. And when we reviewed the literature, there was a suggestion that magnetic bead-based isolation methods recovered more cell-free DNA than genomic DNA. So in this study, this group from Germany showed that the proportion of cell-free DNA isolated in the range of what we believe includes mononucleosomal molecules was higher when the technique was bead-based, whereas the genomic DNA recovery appeared to be very similar. However, these data did not show us whether mononucleosomal or dinucleosomal molecules were favored within that short fragment. And so that's what we set out to do. We compared using healthy donor plasma, both a silica column-based method against a bead-based method. And we wanted to look at both the yield and the integrity and the recovery, and whether the input volume affected either of these measurements. Because certain studies will say a minimum of 5 ml, certain studies will say a minimum of 7 ml. So we wanted to see if that initial input volume affected any of these measurements. And the metrics we looked at were, for integrity, were the length and number of mononucleosomal and dinucleosomal molecules recovered. And then we also looked at the quantity of cell-free DNA per ml of plasma recovered. So in terms of integrity, we performed 15 extractions on the same pool of healthy donor plasmas, and we showed that in 13 out of the 15 isolates, dinucleosomal molecules, as shown here, could be identified in the bead-based extraction method in excess of 300 bases, whereas none of the column-based method showed any measurable dinucleosomal molecules. There was actually a blunting of that shoulder, potentially representing some dinucleosomal molecules, but there was no discrete peak. Next, we showed that the bead-based method recovered decreasing quantity. If you look at quantity of cell-free DNA per ml of plasma, the bead-based method recovered decreasing quantity at increasing volumes, and these volumes range from 2 ml to 20 ml. Whereas the column-based method had a very consistent recovery and very predictable recovery of cell-free DNA. Though bead-based methods appear to always recover more cell-free DNA at every volume. So this loss in cell-free DNA quantity at higher volumes was a very interesting finding we were not expecting. And when we compared the loss of cell-free DNA recovery to genomic DNA recovery, we actually realized that the genomic DNA loss was greater at increasing input volumes, which actually resulted in what you're seeing here on the bottom. If you calculate percent cell-free DNA recovery, which is cell-free DNA divided by cell-free DNA plus genomic DNA, you actually see the percent of cell-free DNA recovery increasing with the bead-based method, and actually also with the column-based method. At increasing volumes. So that at the highest isolation, you can see you can achieve almost 60% purity, what I call purity of cell-free DNA by the bead-based method, whereas the column-based method was still around 30% at the highest proportion. So from these studies, we believe that bead-based methods are enriching for longer cell-free DNA molecules, potentially even enriching at higher, for purity of cell-free DNA at higher extraction volumes. And these studies are still ongoing, and the sequencing data are unfortunately unable to be shared today. So question for the audience, testing to see if folks are still around, is that do you typically use bead-based or column-based extraction methods in your lab? And I will give everyone a few minutes to respond. Or not a few minutes, a few seconds to respond. We'll wait just a few more seconds. Okay, so let's take a look. Wow, so this is very interesting. So about half and half. And then there is a small minority that use another method. So it's very interesting, and this is something you might think about as you start to discover this IG sequencing assay, is that if framework 1 amplicons are important, you may want to optimize that initial DNA extraction process. Okay. So the last portion, we're on the home stretch. The last portion of this talk will be dedicated to interpretation of IG and GS data for clonal tumor-associated sequences. Okay, so first, IG NGS interpretation is largely based on existing PCR and capillary-based methods. So similar to capillary, which is shown in this small inset on the left. Usually we resolve the sequences by amplicon length, which is plotted against signal, right? Signal could be fluorescence signal in the capillary method, or could be reads, right? Read abundance or percent reads on the y-axis. So typically the length is represented as either the absolute sequence length, DNA length, as shown in these two figures, or it could be the predicted amino acid length as shown in the bottom left. In either case, these plots, once plotted against signal, i.e. abundance, you typically are able to identify a dominant sequence. Other groups, like this one on the bottom right, and actually this figure on the top left, other groups have chosen to visualize clonality using the actual VJ genes. So you could look at the VJ usage along with the abundance at the same time. Now, this I want to note a subtle difference between the way my group, which is on the top right, displays our IG clonality data. So when we do the analysis, the amplicons that overlie one another at the same length, even though they're different VJ genes, we do not stack those amplicons. Whereas you can see in the Memorial group and in the Euroclonality group, they stack their amplicons. And the reason we have chosen not to stack our displays is that we have found, as in this case, which is IND stands for indeterminate, we have found that indeterminate clones are a bit easier to identify when the peaks are not overlapping, are not stacked. So that's just one subtle difference. And here are some representative clonal patterns derived from duplicate reactions from a study that we had done. So remember the design is on the top. The first panel here shows you a clonal signal that's actually biallelic. The color codes correspond to the specific VJ genes that are used. In this case, a different VJ combination is used for that second allele. Now, the amplification, obviously there was bias here. We talked about amplification bias previously. There's clearly amplification bias, right? Because these two, while being biallelic in the same cell, from the same cell, do not amplify at the same rate. The second example is a clear clonal monoallelic example. The third is a clear clonal monoallelic where there is actually polyclonal background signals, whereas the previous is in the absence of polyclonal background. The fourth example is what we call an indeterminate for clonality category, where the signal is not strong enough to be definitively clonal. This fifth one is a polyclonal pattern, and the last one, as you can see from the y-axis, is actually what we call a pseudoclonal example, likely due to very, very low input DNA, where it appears to be clonal, but this VJ gene does not reproduce in a replicate reaction. So if clonality can be determined through NGS, how does it compare to PCR? So the Memorial group actually confirmed the concordance of NGS compared to capillary. And they showed that overall, out of 94 samples, there was great concordance. And that the majority of the discordance was actually due to improved clonal identification by sequencing over capillary. Now, they did note that certain samples, this is a smaller number here that were tested by frameworks 1 and 3, certain samples did show a discordance where the capillary-based method was able to identify a clone, but the NGS method was not. Now get ready, we're on to our second polling question, which is, which method do you use or does your lab use for clonality testing? Is it by flow cytometry, by PCR, by NGS, or by another technique? We'll give everybody a few seconds. Now, your lab might use multiple, but maybe you can choose the most commonly used. Okay, so let's see what everybody said. So very interesting. Yeah, so most labs are using PCR-based techniques, although there is a healthy proportion that are using next-gen sequencing and in a smaller number using flow cytometry. I think the major limitation for flow cytometry is obviously the presence or absence of the cells of interest and the need for fresh samples. So if flow was not done up front, then for sure PCR or NGS techniques that can be used downstream on formalin tissues would be much preferred. Okay, great. All right, so let's dive into a little bit more about interpretation. So how do different groups actually arrive at an interpretation of clonal versus non-clonal? Now, currently, there are no clear guidelines, and I'm hoping over the coming years, maybe guidelines will develop just like they were developed for PCR-based, right, clonality. So if we look at next-gen sequencing-based clonality interpretation, there are two main metrics that we can use. What I call read representation or percent reads that tells you your clone percent representation of your clone against all B-cell reads. Then there is a second metric, what I call clonal dominance, which can be calculated as the fold difference between your clone, usually the top 1 or 2 sequences, your top ranked sequence, against a background. You can determine that background as 4th, 5th, 6th, or 7th, or et cetera, ranked sequence. So those are two metrics that you can play with. Now, in order to think about a threshold for calling a clone, we have to actually look at the baseline, i.e. polyclonal samples, and the upper limit of the polyclonal samples. So in this recent Euroclonality Consortium paper, they sequenced approximately 80 reactive lymphoproliferations, and they showed that by these bar graphs, and you can see they're grouped by target So either the framework 3J, the KJ, the DJ, etc. They grouped this by target, and they showed that the top sequence, the most dominant sequence in these lymphoproliferations that were in a polyclonal sample, so in blue, in polyclonal samples in these reactive lymphoproliferations, the vast majority of the dominant sequences fell below 5% of reads. However, you see actually several, right, that are outliers, that in these polyclonal, right, patterns, the dominant sequence could be well in excess of 5%. They also saw the opposite direction, where in green or lime green, they show you the percent reads of the clonal sequences, because there were occasionally they identify clonal sequences in these reactive lymphoproliferations. They showed that these clonal sequences sometimes fell well below 5%, as in this example. So that the percent reads itself as a single measurement is likely not enough, and that the background polyclonal pattern, i.e. the dominance of your right sequence, your dominant sequence of the background, is likely also very important. So knowing these two factors, our group actually tried to develop an automated algorithm to screen Ig and GS data for the presence or absence of clones. So we collected approximately 140 samples from patients with and without lymphoma from 2 different institutions. And I've shown you here the current, what we call gold standard, but really it's the manual or visual review criteria used by either of these 2 sites. You can see that both sites have criteria for clonal, have criteria for indeterminate, and use different measurements. So in site 1, the clonal dominance was used, whereas the percent reads was not used. In site 2, both of these features were used. However, they performed all their sequencing in singlet, whereas the site 1 performed everything in duplicate. So both sites actually had 2 features to optimize, i.e. Site 1 was clonal dominance and replication of the result, and then site 2 was percent reads and clonal dominance. So what we then did with these data was that using these 3 features, we trained several algorithms based on a subset of the cohorts, and we developed several classifiers. The details of the classifiers are not terribly important, but it's to say classifier 1 actually used all 3 parameters, i.e. dominance, percent reads, and repeatability, or what we call repeatability. Whereas classifier 2 only used 2 of them, classifier 3 the other 2, and classifier 4 used repeatability alone. Now the sensitivity, the resulting sensitivity and specificity shown here are quite detailed, because we assess this both on the sequence level, on the primer set level, and on the sample level. But the take-home message here is that no matter the granularity of the analysis, sensitivity matching, so matching high and low sensitivity, or high and high specificity and sensitivity, was achieved when all 3 parameters could be used. So on the sample level, you can see that 100% sensitivity and 88% sensitivity and 100% specificity could be achieved using the most comprehensive classifier, whereas it would decrease if one was dropped out. So this was an algorithm that we developed, which, as you can see, is clearly not perfect, right? It's not yielding 100% both sensitivity and specificity. However, this type of algorithm, we believe, can be used in the future to standardize interpretation of Ig and GS clonality. And that perhaps these types of algorithms can be used to screen your data right before you're visually reviewing it, to reduce the time and effort needed to assess clonality visually. So now that you've heard about several groups doing this kind of analysis in clinical inpatient samples, is there actually a consensus, right, for NGS clonality? Are there criteria? So unfortunately, there is no current gold standard, but you can see that percent reads and dominance are employed by by many, if not most labs. So you can see that 5% also stands out, right? 5% as a minimum also stands out, but there are lots of subtleties here. For example, the Memorial Group actually provided really nice quality control metrics related to minimum read counts for interpretation. They had a category for oligoclonality. The Euroclonality group actually preferred to use dominance rather than percent reads when they were generating their sensitivity and specificity plots. And then obviously our group, we use mostly dominance and repeatability, but labs that are doing things in singlet may actually want to use both percent reads and dominance as measurements. So what we do see from this is that currently there's no gold standard, there's no criteria. However, labs can develop their own, and that hopefully by sharing their approach with others over the coming years, there may be interpretation guidelines that could develop. And what we hope that happened with the Biomed 2 group and PCR, we hope that would happen with NGS, and that the sort of power of NGS, like I mentioned earlier, really comes down to the ability to resolve the sequence. And so it gives you a much more comprehensive view. If we do a comparison of the two methods, we see that many of the factors are similar, such as minimum input, minimum data, clonal dominance, right? So most of the capillary or PCR-based methods are relying on dominance alone. However, where NGS diverges is its ability to give you the exact sequence, right, of your clone at the same time as fragment length. So you see, this now affords you the ability of having an additional measurement of clonal percent rather than just dominance. It gives you the ability to distinguish by allelic versus biclonal proliferations based on productivity of the sequence. Again, because you know the sequence, you're able to determine if something is a pre-germinal center, i.e. immature B cell, or a mature B cell, and therefore obviously have the ability to assess somatic hypermutation. And then lastly, because these sequences, unique sequences are generated, considerations for how to merge when they're the same clone versus subclones must also be considered, and that subclone can be detected by next-gen sequencing. And actually, that is the end of the talk. So in closing, I hope you have a better sense of the next-gen sequencing world of Ig genes, the 4 learning objectives. I hope you've taken at least one tidbit away from each, and that you've seen that this is a very powerful technique that can improve our understanding of both lymphoid malignancies and immune repertoires, which obviously I now talk about, but that I'm hoping in the coming years there would be movements towards streamlining the interpretation of clonal determination by NGS. And these are my references, and I'm happy to take questions.
Thank you, Rena. Well, that's actually all the time we have for today. So we'd like to, again, thank you, Rena, Xian, and our sponsor, InVivoScribe. And if you have a question, please do email it to inquiry@invivoscribe.com. And as a reminder, please look out for the survey after you log out to provide your feedback. And if you missed any part of this webinar, or if you would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.
Thanks, everybody. Bye-bye.